This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
Welcome to A Conversation with History. I'm Harry Chrysler of the Institute of International Studies. Our guest today is Svante Pääbo, who is director of the Max Planck Institute for Evolutionary Anthropology. He's a Swedish biologist specializing in evolutionary genetics. One of the founders of paleogenetics, he has worked extensively on the Neanderthal genome. He is the author of Neanderthal Man in Search of Lost Genomes. He is at Berkeley in the fall of 2014 to give the Forster Lectures on the immortality of the soul. Professor Pääbo, welcome to Berkeley.
Thank you.
Where were you born and raised?
Well, so I was born in Stockholm, Sweden, and grew up there.
And looking back, how did your parents shape your thinking about the world?
Oh, well, maybe my background was a little unusual in that when I was born to my mother, my father was already married and had another family. So we were sort of the secret family. My mother was a chemist, and I think influenced me a lot. My father was a biochemist. He had less of an influence, but he did have some influence in the background. He spent like Saturdays with us and always took an interest in what I did and things like that. But it was really my mother, I think, that...
And he was a Nobel laureate?
Yes, he eventually then got the Nobel Prize.
And she was very supportive of all of your interests as you grew up, and must have been a powerful force in shaping your interest in science.
Yes, and I think one thing that influenced me a lot was that sometime when I was 13 or so, she took me to Egypt, and that's really when I discovered the ancient history that became a fascination to me, as for many young people, I think. And she really sort of encouraged that. But also encouraged, I think, a very sort of quantitative, rigorous attitude to things from her chemistry background.
And did you have an interest in mummies before you went to Egypt, or only after you went to Egypt?
That's really after I went to Egypt. It was such an experience for me to realize that in Egypt you can walk around and sort of old ruined fields where the ground almost is composed of potsherds that are thousands of years old. And there are, these dubious antique dealers that will offer you mummies or pieces of mummies that they at least claim is from pharaonic times. That there was so much things preserved from thousands of years ago really blew my mind at the time.
So it was almost inevitable that you would become a scientist of some sort, or not necessarily? I mean, was your education as a young person very supportive of your interest in science?
Well, I think my interest was then really in Egyptology and archaeology, and I thought I would become an archaeologist and excavate in Egypt, and started studying those things at the university. And then I think it sort of showed that I had a too romantic idea about what Egyptology would be. I thought it would be more Indiana Jones-like things. And it was, at least in Sweden at the time, very linguistically oriented. I ended up studying, ancient Egyptian verb forms and things like that. So then I didn't really know what to do. I pursued these studies for what amounted to almost 2 years.
This is an undergraduate?
This is at the university, yes. And then I think it was probably also my father's influence that I then, when I didn't know what to do, decided to study medicine with an eye towards doing research then.
And was his influence, did it emerge out of conversation or knowing what he did, or
more from diffusion, I think, than really. I mean, both my parents were very good in supporting me in the things I wanted to do and not trying to prescribe anything, which is probably a recipe for disaster with young people.
And you, an idea that emerges in your book, which is a very good account of doing science and the research that you've done, is you have an adventure. You are an adventurer. In other words, there's a little bit of Indiana Jones in you in terms of exploring new frontiers.
Maybe, yeah. I sort of sometimes say to students, if they wonder if they should pursue a PhD or so, is that you should only do it if you really enjoy it, if you think it's fun. You know, we're probably, if we come that far in life, all smart enough to work in an insurance company or in a bank and earn much more money. The only reason to really do research is that you're fascinated by what you do and enjoy it every day.
So after undergraduate work and realizing that you didn't want to just be alone with mummies as an Egyptologist, you decided to go to medical school.
Yeah.
As the career that you would pursue first. And what was it about medicine that convinced you had to do more?
In Sweden, it's still the situation that you rather often go to medical school also when you want to do basic research. So that was really why I started. Then I more discovered as I went along and did the more clinical courses that I also enjoyed seeing patients very, very much, and liked the interaction with people from all walks of life that you get as a doctor. So I had a little mini crisis about should I stay and finish my medical degree and become an MD or do research. And I said, let's try research. I can always come back and finish it later. And that's where it ended. I never came back.
And the research you chose to do, or what you got your PhD in, is
sort of molecular immunology, if you like, how the virus deals with viral infections and interacts with viruses.
One of the interesting themes that emerges is your many careers, in a way, or in your formative years in education, medicine, molecular biology, with this passion for ancient beings and artifacts. You really were drawing on all of these disciplines later in life, in a way. So your education gave you entree to new worlds that were opening up that you then applied to this passion that you had for ancient things.
Yes, and I think it's sort of a good thing with a sort of university system if it allows you to study different things that you can then bring together. It was, of course, quite obvious for me once I started to learn to manipulate DNA and clone it in bacteria, as we did at the time, to say, well, might there not be DNA preserved in all these hundreds and thousands of mummies of humans and animals that I knew were stored in museums around the world and were found in Egypt every year. So I started looking in the literature to see if anyone had done anything, and to my amazement, seemed that no one had tried it. So then it was pretty obvious to say, let's see if this can be done.
Before we pick up that theme, I want to ask you about the skills you think are required to be an accomplished scientist. Obviously, you've suggested learning many specialties in a way. Anything else?
In some sense, I would say that science is a wonderful thing because there are many, many different ways of being a good scientist. There's not just one way. there are those that are incredibly knowledgeable and thoughtful and really think very hard and then come up and do the crucial experiments. There are those that try many, many things. One of them turns out to work out. And of course, one way of doing it is to bring together knowledge that are normally not combined with each other, which might be particularly fruitful to do, especially if you're not incredibly smart. Because, of course, if you're sort of moving into a field where many other very smart people work and say, I'm going to really make a contribution here, you have to be very, very smart yourself. But if you bring things together from different areas, you necessarily don't have to sort of be super smart.
So in terms of temperament, you're suggesting, or you suggest in your book, in a way, be an experimenter, testing not just in the scientific sense, but in the sense of trying different options, but then really persevering when passion comes together with knowledge in a way.
Yes, and I think one sort of driving force was this feeling that it's sort of very frustrating to study history purely from, say, archaeological remains or even texts that you would have from ancient Egypt, but not being able to really know what happened. So there was some feeling there, I think, and still is, that if we can bring some kind of rigor to this, and at least study the population history directly, not by inferring from present-day variation or from old text or from archaeology what happened in the past, but actually go back in time and look at what were the genetic variants in the population that actually existed here 1,000 years ago or 10,000 years ago or even 100,000 years ago, that would be fascinating to do.
It's interesting because what paleontology had before your work and the work of others who were moving in this direction was really artifacts, things that were left by prior civilizations or prehistoric humans or whatever. And whereas what you're really focusing on is an empiricism that relies on the data that lies in the bones in a way.
Yes, and I think even today where one infers population history from studying variation today under certain models and ideas of how things change in populations over time, it is still extremely valuable to actually go back and test those things. And you more often than not, get surprised that things have not been as you had thought in the past.
A theme in your book, again, is the moment where creativity leads to discovery. And there have been several points in your career that one could call wow moments. And one of them is one you just described, namely when the technology came along and evolved to replicate DNA. That was a wow moment for you, which you attached to your passion for studying ancient things. Talk about that moment. I mean, was it something that came suddenly, or did it evolve after much research?
Well, so a sort of a theme all the way over 30 years have then been, can we extract and study DNA from ancient remains? And this then started when one extracted this DNA and multiplied it in bacteria. So that was the first sort of experiments I did in the early 80s. And it was very frustrating because it's a very inefficient process. And you, above all, you can sort of retrieve some DNA sequences and study them, but you cannot repeat your experiments since it's so, to find the same piece of DNA again from that specimen is almost impossible.
And explain that. In other words, in a way, science was using bacteria to ride on the back of bacteria and its processes to replicate DNA.
Yes, so you take your DNA from, say, an ancient bone or from a blood sample. You link it up with a piece of bacterial DNA that then gives it the ability to multiply itself when it's introduced into bacteria. So you use the bacteria as a copying machine, if you like, to make thousands and millions of copies of these small pieces of DNA. But it's a random process. You just happen to pick up some molecules, and it's very hard to then find the same molecule again. So, and as biology is in general, really, it's sort of driven by technological advances. So then came the polymerase chain reaction, which was an invention by Kary Mullis here in the Bay Area. That is a possibility in the test tube to multiply a predetermined piece of DNA, that you decide which piece you're interested in by synthetic pieces of DNA that you sort of use to make many copies of it. And it was obvious from day one when I heard about that thing that this might be ideal for studying ancient remains. So that's then what I started working on, first back in Europe, and then over almost 4 years as a postdoc here at Berkeley with Alan Wilson, which was a fascinating time because suddenly we could go back and actually reproduce our experiments. So I could amplify, for example, a piece of a moa, an extinct flightless bird from New Zealand, and then repeat it again from the same individual and from other individuals to really make sure that the DNA sequences we got were correct. Up to that, with these bacteria, one had never really been able to be sure if this was a contaminating piece of DNA that actually came from me or some museum curator or something like that. And in many cases, it actually were in the earliest experiments.
You quote Gerard Diamond as saying, old specimens constitute a vast, irreplaceable source of material for directly determining historical changes in gene frequencies, which is among the most important data in evolutionary biology. So that description, in a way, is what came to be, really, by your work. And interestingly enough, but all this time you're keeping this passionate focus on ancient things.
Yes, it's really this thing of going back and saying we should try to catch evolution red-handed, if you like, to be able to actually see what was in the past.
Now, in the case of ancient DNA, there are all kinds of problems, and I want to talk about them. You touched on it a moment ago. So really, it's not as if, okay, let's take a DNA sample from everybody who's living, who's in the room with us. Let's talk about go into more detail with the problems with ancient DNA. The first problem is really the contamination that you talked about.
Yes. What really became clear over the first years of work with this was that the DNA in an ancient specimen, even if it's just a few hundred years old actually, is always degraded to short pieces. It's chemically modified, which make it hard to make copies of it. And there's often a very, very little of it there. So in the beginning, when we did, or I did experiments then just on the lab bench in my normal lab, extracted from an ancient mummy, cloned it in bacteria and found some human DNA. In reality, that was almost always DNA from myself or from some other people in the room. It turned out, for example, that dust in a room like this where people move around is to a big extent small skin fragments, that actually each such little dust particle can contain much, much more DNA than what there is in a gram of tissue from an ancient Egyptian mummy, for example. So gradually I sort of came to be more and more paranoid, if you like, about contamination. Separating this type of work in a separate room, where you have UV light at night to destroy DNA, you wash everything in bleach, started dressing in protective clothing that you could discard after each experiment. we started saying that you can only go in there in the morning when you come to the laboratory. You should not have entered any other laboratory where there are large amounts of DNA being handled. And step by step, one slowly got this under control.
And the pieces were very short that you were
Yes.
able to extract from the ancient material.
Yes. So whereas, say, from a blood sample from someone today, you can easily retrieve 10,000, 20,000 base pair long of these letters in the code, long fragments. Here we have things that are 50, 60, hardly ever up to 150. base pairs long.
And through time, there are a lot of chemical reactions acting on the bone fragments, say, that decompose the DNA. Water,
Yes,
for example,
yes.
is one of the things.
Yes.
Yeah.
So DNA is a very hydrophilic molecule, so it sort of absorbs water molecule around it. So even in an environment that we think is dry, there is water around the DNA molecule. And that leads to reactions. Some of them break the DNA to smaller pieces. There's others that modify one of the 4 letters in the genetic code, the C's, so that when we study them, they turn up as, they look as another base, another T. So there are certain types of errors that you can actually then sometimes use to your advantage to say, I really believe this DNA is old because I see these types of modifications in it.
Mm-hmm. And so the cleaning process, help our audience visualize the cleaning process. It's one we see now with epidemics emerging, the way the personnel treating those suffering from the illness, the outfits they wear, the decontamination process we see there is what you brought to the process of looking at ancient DNA.
Yes, so it looks on the surface of it very similar. You sort of dress up in a sort of outside the room you work in this special clothing, you work in a hood. But what we really do is protect our experiment from ourselves, from the DNA we would shed. But it looks very much like, say, a chip factory where you make computer chips. So there's sort of, the air is filtered, so there's no dust there. There's high pressure in the room, so the dust cannot come in, but the air sort of flows out of it.
Now, another problem that you encountered in this work is where are the bones, basically. And once you know where the bones are, getting the various bureaucracies at play that control the bones to give you some piece of the specimen. Talk a little about that, because that was a as you describe it in your book, it's Byzantine maneuvering and political instincts were required on your part.
Yes, that's of course very different in each place and in each country. And my experience, it's generally easy to deal with the people who have actually excavated and discovered the things. And of course, I respect very much that if you discover the bone, you feel a sort of scientific ownership of it, which is totally appropriate. What often happens, though, is that there are museum curators that sort of inherit the keys to the cupboard from things that were found maybe 50, 60, 70 years ago, and then controls it as if it were their private property. And that's what sometimes makes me a little upset. So then it's of course, they need of course to balance the sort of, that the specimens are often valuable and should be preserved for the future against the scientific knowledge you could gain by actually studying them. But it's of course also the case that we can often study fragments of bones that have very little morphological value. For example, the bones we first used to make the first version of the Neanderthal genome were from fragments that one could not for sure tell if they came from a cave bear or from a Neanderthal, for example. So then there is obviously very little value from studying these bones morphologically. And then it can get frustrating sometimes when it's still hard to get access to these things.
In several cases, the bones were actually in the countries that were formerly part of the Eastern Bloc.
Yes.
The first important set, I think, were in East Germany when that country was still divided, and then there was a real find in Croatia.
Yes, yes.
Which probably made the bureaucracies even worse, to the extent that they were remnants of the old Soviet system.
Yes, so many of these things have been found in the former socialist countries. And of course, the structure of how science work there is partially different. Say the academies of sciences are very influential and things like that. So.
Talk a little about science itself, because it seems that on one element of science is cooperation, international. You're working with scientists all over the world. On the one hand, as you mentioned, you were here in Berkeley for several years. But on the other hand, competition, basically.
Yeah.
Who's going to get to the press first with whatever revelations emerge?
Yes, so there is sort of this tension all the time. And I think competition to some extent is good, right? It sort of keeps you on your toes and makes things go faster than they would otherwise do. But above all, I think what is so great with science is the cooperation. The fact that sort of for analyzing the genomes we now produce, we work together with people all over the world, but particularly here in the US, actually, particularly, say, Monte Slatkin's group here at Berkeley and David Reich and his collaborators at the Broad Institute and at Harvard and at many other places. And that you can really, with today's technology too, work very closely together. You can have video conferences every week and actually really have the feeling you work together towards a goal. And in this project, it was particularly nice because everyone involved really had the feeling this was a unique first view on an extinct forms of humans. So it was amazing how everybody pulled together and worked unselfishly to sort of arrive at the things.
You were fortunate in the sense that you were invited to be part of an institute that was being founded that focused on evolutionary biology, the Max Planck Institute. Talk a little about that, because in every great scientific breakthrough, there is a team that's put together, and you were fortunate enough to be part of a process putting a new team together that drew all sorts of disciplines together on the one hand, but also were focused not on disciplines, but on the particular problem that interests you.
Yes, I was sort of very, very lucky to be in Germany, happened to be in Germany then at a time when there was a political decision to build up science in East Germany, and particularly then Max Planck Institutes, which are focused on basic research, to the same density in the East as there was in the old West German part of the country. And it was then the will to focus on things where German science would be particularly weak. And one such area was, of course, anthropology, because of the terrible history in Germany with the Holocaust. So there had been a sort of predecessor organization to the Max Planck Society before the war, where many of Max Planck and other very excellent scientists had worked. But it had gotten very involved with the Nazi crimes, had an institute in anthropology that was involved in the Holocaust. So since 1945, one had not touched the subject, really, for very good reasons. But now it was a feeling maybe one should do it again. And I think it was maybe even a little easier for me as an outsider working in Germany to say, we cannot have sort of what happened in the past dictate what we can do now. We have to be able to go forward. And once one sort of made that courageous decision to found an institute of evolutionary anthropology, turned out to be almost an advantage to have no traditions to fall back on. That we could sort of sit down, the people that were involved in starting this institute, and say, how would we today start it if we do it from scratch? And what we ended up doing was sort of focusing it on a question rather than on discipline, to sort of the question, what makes humans unique in comparison to other organisms? So as long as you work on this, and as long as you work empirically, was the other sort of litmus test, you could work there no matter where you came from. So there are comparative linguists, for example, that study what's unique about the human language. There are primatologists that study the behavior of our closest living relatives, chimps and gorillas and orangs, in the wild in Africa. There are comparative psychologists that do experiments in the zoo in Leipzig on the apes, the children during development, and try to do the same experiments in human children as they grow, to find differences and similarities in how they evolve. And there's paleontology that then excavate remains of Neanderthals and modern humans, and there is genetics, where we then focus on comparisons to our closest extinct relatives, the Neanderthals then.
So in a way, metaphorically, this is about building bridges between disciplines, and then building a bridge to the past where you can come to understand the leaps that our ancestors, the pre-human ancestors, made to make us what we are today.
Yes, and I think the problem is always to get these bridges between disciplines to really work, to really understand each other. And I think a critical thing there that sort of made this work as well as it did was really to focus on scientists that do empirical work. Because I can actually understand what a linguist does, although I'm not a linguist at all, if they then just take the time to explain to me what the data is, what the hypothesis, the test with this data is, and one can then have a fruitful interaction.
Science, as you describe it in your book, is a social process. And on the one hand, there is the process of interaction with these colleagues that start in other disciplines. But on the other hand, there is the interaction with your own group, and bringing to that group problems that are emerging as you're doing the research, and through conversation and discussion and criticism, really finding the way forward.
Yes, I think it's certainly a social process, science. And really in our case, the crucial thing is really the group. The group as an entity is much, much smarter than any individual in that group. And my role is then often just to sort of catalyze that lots of ideas are put forward in weekly discussions we have about the projects, and sort of maybe sort of identify some ideas that come out as particularly worth to pursue. But it's really, when the group works well, it's almost impossible even to say who comes with the idea, because it's a process that you do together.
And there also is another aspect here that has both a positive and a negative quality, which is that there is the word conventional wisdom about this domain and what we understand. And when you're doing innovative work like you were doing, then you have to overcome the obstacles of the conventional wisdom about what we know.
Yes, I think that almost one quality that is good in a scientist is a little bit of an anarchist, to be able to sort of almost take a delight in questioning this sort of received wisdom and hope to overthrow it. That you cannot believe in authority or believe what is in the textbooks. You must sort of be able to go back to first principles and say, how do we actually know the thing that is there in the textbook? Could it be wrong? Could the whole world be wrong and we be right, actually? So there is some delusion of grandeur or what you like there. That sort of, to be able to instill in the students and people in your group this feeling that you can actually might be right and the whole world might be wrong.
You say at one point, I am driven by curiosity, by asking the questions, where do we come from and what were the important events in our history that made us who we are. So now that we've cut through the brush, talked about all the obstacles, talked about the impact of technology and the problems with the DNA, help us understand what you and your group were able to achieve with regard to Neanderthal Man.
Well, so there had, of course, been a debate for decades in paleontology what really happened when modern humans came out of Africa, starting around 100,000 years ago and really seriously spreading from around 50, 60,000 years ago. When they met Neanderthals, what happened? Did one mix with each other or not? Is there a contribution from Neanderthals to people in Europe today? And it had been a long, long debate with really no clear resolution of that. And when we then finally got a version of the Neanderthal genome, one could then directly compare that genome to people living in different parts of the world today. And what one found was that there were pieces in the Neanderthal genome, or pieces in the genomes of people today that were very close to the Neanderthal genome. And you found those pieces in Europe, in Asia, and not in Africa. And by various sort of analysis, particularly then by people like David Reich and Nick Patterson at Harvard and Monty Slatkin here, one could sort of show that the only way really to explain that is that one mixed with Neanderthals rather recently, around 50, 60, 70,000 years ago.
That is humans.
Humans,
yeah.
Yes, modern humans mixed with Neanderthals. And so that in the order of 1 or 2% of the genomes of everybody outside Africa come from Neanderthals. So it was clear that one did mix, but it was also not that one found that Neanderthal contribution only when Neanderthals had existed in Western Asia and Europe. Found it even in China, in Papua New Guinea, in Native Americans. So the model that has come out of that is that when modern humans come out of Africa, they rather early mixed with Neanderthals. And those modern humans may then later, here and there, are mixed with Neanderthals again, but overall they become the ancestors of everybody outside Africa. So that no matter where people come from, if they are from outside Africa, they have at least about a percent of Neanderthal DNA.
So what surprised you when all the data came in and all these procedures had been implemented to find pure DNA was this, you were surprised and your team were surprised by this 2% of Neanderthal DNA in the human genome?
Yes, I was biased to think it had not happened because we had earlier in the 90s studied just a tiny part of the Neanderthal genome, the mitochondrial genome. Which is inherited only from mothers to offspring, and it's a very small part of the genome. And there we had found no evidence of mixture whatsoever. So I was biased. I sort of knew, of course, that wasn't the full story, but I was biased to think one had not mixed. So for the longest time, I sort of thought there might, after all, be some mistake in what we do. We might have some contamination we overlooked or so. But in the end, it was sort of completely clear that that was not the situation.
So you had to confirm by analysts outside of your group that what you were doing and the conclusion you had reached was right.
Yes, I mean, the theoretical analysis was large part done by these other groups I mentioned, and actually by different approaches. There were at least 3 different independent approaches that came to the same conclusion.
What do you think this means that our ancestor was in some ways and in some small parts Neanderthal Man. Do you draw any larger conclusions from that, other than really impacting the theories that were out there about the movements of people in prehistoric times?
Well, so it's beginning now in the few years since we've done this, it's already becoming evidence that some of this contribution from Neanderthals have had some sort of real effects. So for example, there's a group at Stanford, Peter Parham has shown that some genes that are involved in regulating the immune system, transplantation antigen genes, have been contributed from Neanderthals and relatives of Neanderthals in Asia to present-day people. And one can easily imagine that that has to do with that these modern humans come out of Africa, meet these groups that have lived for several hundreds of thousands of years in other environments and have adapted to infectious diseases there. When they then mix a little bit and genes come over that are advantageous to fight those infections, they rise to high frequency or are advantageous. There is a risk variant that was found this January for type 2 diabetes, the type of diabetes you get in old age that is high frequency in Asia and in Native Americans that also come from Neanderthals. One can easily imagine that that is some adaptation to starvation, to store energy better. And today we get diabetes when we store energy too well, when we eat well all the time. And there is a recent paper here from Berkeley, from Rasmus Nielsen's group, that have showed fascinatingly that people in Tibet who live on the high plateau in Tibet and are adapted to living at a low oxygen tensions there, have gotten a variant that helps with that ability from relatives of Neanderthals that we have discovered, these Denisovans in Asia. And 80% of Tibetans have that variant today. So one can put this sort of, it's beginning to emerge a picture of adaptive introgression, as we say, saying that genes come over from these other groups, some of them are advantageous and will then rise to high frequency and actually have its impact.
Another place where this has great implications for is the study of the brain and how it functions and how it evolves. And as part of your collaboration with your colleagues at other disciplines, I guess you learned and focused a little on what is called the FOXP2. Tell us how genes became important in understanding a small group of living humans and what it might point to with regard to the connection with Neanderthal.
Yes, so there's one thing, as we discussed so far, then what came over from Neanderthals to people outside Africa. But it's another set of questions that is even more fascinating to me almost, and that is to say, in what parts of our genome do all humans today, no matter where we live on the planet, in Europe, in Africa, or in Asia, have something in common that's not there in Neanderthals, and where the Neanderthals look like the chimpanzees and other apes. So those things that are actually unique to all present-day humans, that define modern humans genetically, if you like. And that list of things is not very long. So it's a bit over 30,000 changes in our genome. So to just make that, expand on that a little bit. So whereas, say, I differ from you or from a Neanderthal at, say, 3 million positions in the genome, if we make the requirement that we all today should share something and be different from the Neanderthal and the other apes, it's just 30,000 such changes. So a dream is that among these will hide some of the changes that are crucial for what made modern humans so special. That we expanded from being a few 100,000 people that were like the Neanderthals to being 7 billion today, that made us able to develop a technology that today is incredibly different from 100,000 years ago, to develop art, music, many other such things. Of course, we don't know what those changes are, but they presumably have something to do with how the brain functions. So there's special interest now. We and others in the world are particularly looking at this list of things, thinking about, are there things there that might be important for this?
And in the case of the FOXP2, there was a family that had difficulty speaking.
Yes.
And so and you learned about that from a colleague, I guess.
Yes.
And then it becomes interesting to see if any of these differences in the genome can be attributed to that. Is that, yeah?
Yes, so this gene FOXP2 is one of very few, or the best gene that we know have specifically something to do with language production in humans. Because Tony Monaco in Oxford found this gene in a family with a severe language and speech problem. And that gene turns out to encode a protein that is very conserved among all mammals, but in humans have 2 amino acid differences in the encoded protein compared to all other apes and primates. So we were, of course, very interesting when we got the Neanderthal genome and this genome of the Denisovans in Asia to see, did they share these changes? And they did. So they look like us. So these are changes that happened before our split from the Neanderthals, but they are very interesting, of course. So one thing we have done, which is a model for further work on this type of human-specific changes, I think, is to put them into the mouse. So you then create a mouse that now makes a human version of this FOXP2 protein and study that mouse. And amazingly, that mouse actually vocalizes slightly differently. There are some differences in how it peeps, subtle, small differences. And you can also study how the brain functions. So how electrical signals are transmitted in the brain, particularly part of the brain that has to do with learning muscle motor behaviors. And there's some recent unpublished work that even suggests that these mice are actually a bit quicker in automating muscle movements. A little bit like when you learn to bicycle, say. When you think about how you bicycle early on, it's very difficult. When you automate it, you get very good at it. And it's a little bit like articulation when we learn to speak. Initially, as kids, it's very difficult to form the words. And when you get sort of in our age, it comes rather easily to you. So you can speculate, of course, that these changes may have something to do with this ability to produce articulate speech. And there's some evidence then for it from the mouse. And I think that's how you need to start working with many of these human-specific changes. Try to find animal models that give you some indications of it. Maybe introduce them in stem cells to in vitro, differentiate cells, nerve cells, say, and see how they function.
There's an irony here because you left medicine to go back to the past to make an important discovery, and now we're getting feedback back into the future in medicine and understanding the genetic basis for some ailments. Yeah,
Yes.
Fascinating. Let's briefly talk about this discovery of the Denisovans,
Denisovans.
Yes. Because that came late in this process that we've discussed, and this was just a eureka moment. So this is another group of people that was found in Russia, but who their virtue was the DNA was really amazingly intact.
Yes, so we're very lucky to work with people in Novosibirsk, particularly Anatoly Derevyanko, sort of very influential archaeologist in Russia, who excavated many sites in Siberia. And in 2008, his team had found a tiny little bone in this cave called Denisova Cave in the Altai Mountains in southern Siberia on the border to Mongolia and China. And this was a piece of a last phalanx of a pinky. And we got a sample of that. And it turned out that the DNA there was rather a lot of DNA relative to bacterial DNA in there. So we were able then in 2010 already to sequence a large part of that genome and compare it to the Neanderthal genome and present-day people's genomes. And we were very, very surprised. I was sure that this individual would be either a modern human in that area or a Neanderthal, sort of at the eastern extension of the Neanderthal distribution. And it turned out to be something else. So it has a common ancestor with Neanderthals, but very far back, in the order of 300,000–400,000 years back, much older than any divisions between present-day living humans. And then a long independent history. So these are some sort of Neanderthal relatives in Asia. So we needed to come up with a name for this group. So we said, just like Neanderthals are called Neanderthals after Neanderthal site where they were first found, we call these guys Denisovans after the Denisova cave where they were first found. And it turns out that they have contributed also to present-day people all over Asia to a small extent, but particularly in the Pacific. So people in Papua New Guinea, Aboriginal Australians, contain up to 4–5% of DNA from these Denisovans.
And in this cave, you found evidence that Neanderthals were there, this group was there, but also humans were there at one time.
So we have also found other bones in the cave that come from Neanderthals. So this is an area where at sometimes Neanderthals have lived and sometimes Denisovans have lived.
In this work, and as you reflect on your work, have you done any thinking about the implications of all this for the position, the place of humans in this whole picture of creation in terms of who we are and where we're going?
Well, of course, it is fascinating to me in a way that these groups of humans, Neanderthals and Denisovans, who are not here anymore, that are extinct, that they still live on a little bit in many of us today if we have our roots outside Africa. So they're not totally extinct, if you like. They have contributed a bit to us. Of course, it's fascinating also to say that Neanderthals were here just, say, 3,000 generations ago. It's not that long ago. So sometimes I like to sort of speculate and say, what if they have made it 3,000 generations more? What would that really mean for our sort of ideas about human uniqueness? Would we, if you're sort of pessimistically minded, we would say we would just experience even worse racism against Neanderthals, and we would even worse than what we experience among us today, because they were truly a bit different from us. If you're more positively inclined, you might say, if we had other groups of humans here that were a bit different, better in some respects, but also less sophisticated in others, maybe we would not be able to make this enormous dichotomy that we do so automatically today between humans and animals, that we would see that there's more diversity to life than that. Who knows? It's sort of just something you can speculate about.
One final question. How would you advise students to prepare for a future if they're interested in science and are fascinated by the potential breakthroughs that you just described to us? How should they think about their own future and preparing for a future in which they are part of this scientific discovery?
Oh, it's sort of a difficult question because we cannot anticipate the breakthroughs of the future, of course. But I would, of course, say, follow the things you are fascinated by, because it really comes rather automatically that if you really like something you do, you tend to be good at it. And just get a rigorous training, at least in one discipline, that you really know the basics of it. And then you can bring in other types of knowledge too.
Well, on that note, I want to thank you very much for taking us on this intellectual journey and helping us understand the remarkable discoveries that you and your team made. Thank you very much.
Thank you. Pleasure.
And thank you very much for joining us for this conversation with history.